Hello, my name is Ryan Maloney, and I am the Chief Research Advisor for Zerona, the manufacturer of the Zerona, a non-invasive body contouring device. On behalf of the entire Zerona team, we would like to thank you for taking the time to better understand this unique therapeutic approach for non-invasive body contouring. The purpose of this presentation is to elucidate the fundamental histological and clinical evidence in order to demonstrate the true clinical utility of this application. Although this presentation will probably not address all of your questions, it should, however, establish a firm foundation regarding the efficacy and safety of this technology. The basic features of Zerona include the utilization of 5 adjustable, independent diode lasers, all emitting 635 nanometers, a wavelength which resides within the red visible spectrum. The adjustable diode arms of the Zerona allows the device to treat a diverse patient population, as the diode arms can be extended or concentrated depending on the patient's specific treatment needs. All application modes are preset and require no adjustments prior to administration. The output intensity of 17.5 milliwatts per diode laser labels Zerona as a Class 3B laser device. Therefore, it must be sold to a physician in the United States. Another important feature for Class 3B devices includes the significantly reduced risk of an adverse event. In fact, with tens of thousands of patients treated with Zerona, not a single adverse event has been reported. A unique feature of Zerona includes the non-contact delivery mechanism, in which the device makes no physical contact with the patient, a stark contrast to the majority of other body contouring devices. Another distinct element is the operator-independent treatment, meaning once the device is activated, the clinician is not required to remain in the procedure room, limiting the amount of time dedicated to the operation of Zerona. From the patient's perspective, this allows them to receive the Zerona treatment in the privacy and comfort of the room. And for a patient who has perhaps developed anxiety regarding their body image, this can be an important feature. This image accurately illustrates the innovative delivery mechanism of Zerona. Here we see the device positioned in a concentrated arrangement, treating the patient's abdomen only. Before we move forward, it is important to understand that the results discussed in this presentation were collected following the concurrent treatment of the waist, hips, and thighs, in which the 5 diodes were placed in a manner where light energy was effectively delivered across the waist, hips, and thighs. However, the Zerona is not restricted to that treatment approach. In fact, the device can be concentrated over a specific anatomical area, such as the abdomen only, depicted here, facilitating a greater circumferential reduction in one specific area. It is necessary to better define Class 3B lasers, as the common perception of laser therapy includes the ablation or destruction of tissue. And in fact, for a majority of laser devices saturating the cosmetic and medical communities, this would be a correct generalization. Most therapeutic lasers utilize a photothermal mechanism to produce the desired clinical outcome. Photonic energy is applied to elevate tissue temperature with the intention to promote cell apoptosis or program cell death. In response to tissue ablation, an infiltration of immunological components results in the release of growth factors, proteins, collagen synthesis, and proliferation of new tissue. Delivering light at these energies, of course, can give rise to significant adverse events. This is not the mechanism of action of Zerona. In fact, the mechanism of action for Zerona is synergistic with the agonist effect observed for most drugs. When a drug is introduced into the body, the specific molecular conformation will recognize and bind with a druggable target, which can include a receptor or enzyme. Once bound, a secondary biochemical cascade is triggered, which is responsible for modulating gene expression, protein synthesis,
essentially impacting the functionality and behavior of the cell.
All of this occurs with the patient unaware the drug is working. He or she will not feel this reaction. They will only observe the desired clinical outcome. Zerona operates under a similar agonist mechanism. Here we see in this image the depiction of low-level lasers' effect on the cell's function. Zerona targets a photoreceptor, activating the receptor via the absorption of the delivered photonic energy. Upon activation, a secondary biochemical cascade is induced, which can modulate gene expression, protein function, essentially just like the drug, impacting the functionality and behavior of that cell. Please note that the stimulation of the photoreceptor results in the synthesis of reacting oxygen species, as an important molecule believed to be responsible for the underlying mechanism of Zerona, which we will discuss shortly.
The photoreceptor targeted by Zerona is cytochrome C oxidase, a terminal enzyme of the electron transport chain which participates in the complex biochemical chain responsible for the production of adenosine triphosphate. Based on the structural conformation of this enzyme, it has the ability to absorb light energy at defined wavelengths. Since the Zerona must stimulate the photoreceptor found within adipocytes, some questions have been raised whether this defined parameter of laser energy can penetrate that deep. However, a quick look to the literature and we can quickly find peer-reviewed published articles that have determined lasers with similar output parameters to that of Zerona are able to penetrate the skin barrier and stimulate subdermal tissue. Mathematical equations reveal that per every 1 millimeter of skin thickness, approximately 15% of light energy is absorbed. With the Zerona emitting nearly 45 billion photons per second per diode, with the average skin thickness of the abdomen approximately 4 millimeters, it is estimated that nearly 27 billion photons are able to reach subdermal tissue. Before discussing the effect Zerona has on the adipocytes, it is first important to discuss some basic physiologic features of the adipocyte. After puberty, we find the number of adipocytes remains static throughout an individual's lifetime. It is under rare circumstances that we see the formation of new fat cells. An important responsibility of the adipocyte is to store lipids which are encapsulated by the cell cytoplasm and bilipid membrane. This can result in a nearly 1,000-fold expansion in volume and a 20-fold increase in the cell's diameter. Hypertrophic, or enlarged adipocytes, results in tissue expansion, giving rise to the overweight or obese appearance. Think of the stored intracellular lipids as the load-bearing columns for any structure. Those columns give rise to the structure's shape and volume. When those columns are removed, that structure or room will simply collapse. This simple analogy can be applied to the fat cell. By removing the intracellular lipids, the voluminous structure of the adipocyte will be lost and the cell will collapse. That is the goal of Zerona, to remove the very component responsible for the nearly 1,000-fold increase in volume within adipocytes, restoring a lean and viable adipocyte state.
The underlying mechanism of action for Zerona proceeds via a biochemical process known as lipid peroxidation. It was mentioned earlier that the stimulation of the photoreceptor results in the generation of reactive oxygen species. When increased, ROS can react with the bilipid membrane of the adipocyte, resulting in the formation of transitory pores or temporary openings. This disruption in the protective barrier allows the intracellular lipids to exit the cell, resulting in adipocyte collapse. Therefore, the defined parameters of laser energy delivered by the Zerona stimulates the photoreceptor, which results in the upregulation or increase in ROS, in turn inducing the biochemical reaction known as lipid peroxidation, resulting in the formation of the transitory pores within that protective barrier. This time-lapse footage illustrates the formation of the transitory pore immediately following Zerona.
We were able to prove this outcome at several research facilities throughout the world. The series of images we are presently reviewing were taken at the University of Chicago. The upper left image depicts hypertrophic adipocytes prior to the treatment of Zerona, and the lower right corner images shows the control sample at 120 minutes. We are able to observe the preservation in the structural integrity for the non-treated controlled samples. The series of images, starting in the upper right corner and ending in the bottom left corner, reveal that subsequent to Zerona, a transitory pore is formed, promoting the liberation of the intracellular lipids. Further analysis via scanning electron microscopy and transmission electron microscopy exhibit the loss in volume for Zerona-treated adipocytes. These images represent the foundation of Zerona, how the absorption of laser light can modulate the structural integrity of the cell by releasing the intracellular lipids. The next logical transition was to evaluate whether the findings observed in the basic in vitro studies could be duplicated in vivo. A study was performed using MRI T1 and T2 sequences to assess radiological changes following a single Zerona administration. When comparing the pre-procedure image, we can observe defined septae, the unique fibrotic encapsulations of adipose tissue, a characteristic not observed in the post-procedure image. Post-procedure sequences exhibit a coalescence of tissue, indicating a structural modification in the subcutaneous fat layer. With radiological changes observed nearly 5 centimeters within the subcutaneous fat layer, this study demonstrates that the skin barrier has little effect on Zerona's ability to promote the release of intracellular lipids within an in vivo model. A separate ex vivo study was performed to validate the findings of the previous studies. Abdominoplasty sections were divided into control and test samples, and a series of punch biopsies were performed. These samples underwent adipocyte cell staining analysis and confirmed that sections treated with Zerona expelled intracellular lipids and collapsed. The aforementioned studies elucidated the effect laser has on adipocyte integrity, but did little to explain how a slimming event can transpire in as little as 2 weeks. When we really think about what we have discussed thus far, all we have proven is that the fat moves from inside the cell to outside the cell. It must displace in order to produce the clinical slimming. The Zerona, as a non-ablative technology, will not disrupt the capillary beds or adjacent vasculature. Therefore, there is no possibility for the released lipids to immediately enter the circulatory system. Thus, we will not see a transient rise in serum triglyceride levels. The only other explanation for the slimming observed is the removal of the fatty material via the lymphatic system. In fact, a major responsibility of the lymphatic system is to regulate the amount of interstitial fluid. In principle, we are elevating the interstitial fluid level, in turn, activating the lymphatic system. Lymphatic vessels will remove the lipid debris, transporting it to sentinel lymph nodes, or the closest network of lymph nodes. Lymph nodes contain the immunological agranulocyte, macrophage, which secretes lysosomal acid lipase, an enzyme which catalyzes the hydrolysis of triglycerides into non-esterified free fatty acids. This mechanism is very similar to lipolysis, which is the hydrolysis of triglycerides catalyzed by hormone-sensitive lipase producing free fatty acids which are deposited into the circulatory system to be used as energy. A requirement by the Food and Drug Administration was the completion and submission of fasting lipid panels for subjects to ensure that no elevations in serum triglyceride levels were documented. Those data demonstrated no elevation in triglyceride levels, indicating the removal of the fatty material must degrade the triglycerides prior to the circulatory system. Like most therapeutic strategies, the complete mechanism of action for Zerona is not yet fully understood, but we do remain steadfast to fully elucidate this mechanism. Once the fatty acids are deposited into the circulatory system, there are numerous fates which include beta oxidation, phospholipidogenesis, or even triglyceride resynthesis. We will discuss the latter outcome shortly, and the data we have acquired to dispel that mechanism as a primary fate of the Zerona-liberated lipids. To prove the efficacy and safety of Zerona, a Level 1 clinical study was performed. It is important to understand that the term clinical study is often used to demonstrate to the general public that the product has been validated to some degree. We must understand, however, there are numerous types of clinical studies. There is only one type, however, of clinical study able to evaluate the effectiveness of a therapy, and that is a Level 1 randomized controlled clinical study. A majority of cosmetic devices fail to apply these rigid clinical study parameters when evaluating their devices. In fact, Zerona remains one of the only devices to have been examined in a Level 1 clinical investigation. The clinical study to evaluate the Zerona was a placebo-controlled, randomized, double-blind, multi-centered clinical study performed in the United States. 67 participants were enrolled, with 35 randomized into the treatment group and 32 enrolled into the sham group. The sham device in this study had an identical appearance to that of the active device, and even emitted a rich red glow. So in the event patients were compelled to lift off their blindfolds, they would not have been able to differentiate between the active and sham devices. In addition to the defined study parameters, patients were required to sign an affidavit, confirming they would adopt no lifestyle or dietary changes or participate in any programs that potentially could construe the results. In order to monitor patient adherence, subjects were required to keep daily dietary journals, recording daily caloric intake and participation in physical activity. If any deviation from normal habits was observed, that patient would have been removed from the study. Fortunately, all patients complied with study protocol. Treatment was administered every other day for 2 weeks, for a total of 6 treatments. Each treatment was applied for 40 total minutes, 20 minutes anterior and 20 minutes posterior, with the simultaneous treatment of the waist, hips, and thighs. All statistical analysis was performed by a third-party group. Finally, for any clinical study, it is important to establish a primary outcome, essentially a bar in which you hope to hit. And the number of subjects hitting that bar in that treatment group will determine the efficacy of the application. For most products, that bar is established by the manufacturer, which may not be meaningful. Prior to the commencement of the study, Erconia submitted the trial to the FDA in a process known as pre-IDE. During this process, the FDA clinicians determine the primary clinical outcome based on their extensive literature review and what they feel would be meaningful. It was the FDA that determined a circumferential loss of 3.0 inches or 7.62 centimeters across the waist, hips, and thighs in 2 weeks would be meaningful. Following the 2-week treatment administration phase, test group subjects exhibited a 3.52 inch or 9.1 centimeter reduction in just 2 weeks compared to a 0.68 inch or 1.68 centimeter reduction for the sham group. The difference between the two groups was statistically significant at a p-value of 0.001. Again, these results were achieved in 2 weeks, not months, and without any reported side effects. An additional measure in the study, but equally important, was to evaluate the patient's perception of the results by having all subjects complete a body image satisfaction survey. For 70% of test group subjects, they were either satisfied or very satisfied with their results compared to just 26% of patients in the sham group either being satisfied or very satisfied. Conversely, 36% of sham group subjects reported a very dissatisfied rating compared to just 3% of test group participants dissatisfied with their outcomes. In addition to the satisfaction survey, we had patients report comments regarding why they reported the level of satisfaction. For test group subjects, comments included, "Clothes were fitting better. I went from a size 14 to a size 10. Whereas sham group subjects had comments including, "Could not notice a difference. Clothes were fitting the same." For the sake of time, we will not be going through in detail the description of the images we are presently looking at. But please understand that your sales representative can provide you with thousands of before and after images. This before and after image, however, illustrates appropriately what a typical circumferential reduction of approximately 3.5 inches looks like. This image also represents a great strategy to put in proper perspective for the patient what the circumferential reduction means. This young lady wanted to fit in her favorite pair of jeans. The clinician took the pre-procedure image with her wearing her jeans, and it was apparent that the expansion in adipose tissue resulted in the protrusion of her adipose tissue over the jeans. After 2 weeks of treatment, the patient was asked to place those same jeans back on and dramatically revealed what circumferential reduction of a few inches really meant from a visual perspective. In addition to the Level 1 clinical study, we continued to evaluate the Zerona, performing retrospective studies. This particular study assessed 567 subjects receiving treatment across their waist, hips, and thighs. Early on in this process, we received some criticism that the results were simply based on the redistribution of fluids. And in order for us to successfully address that criticism, a secondary parameter in this 567 subject study included the circumferential measurement of systemic, non-irradiated regions, including the neck, arms, breast, and knees. Theoretically, if we are in fact just redistributing fluids, patients should have shown a circumferential gain in these areas. However, the opposite outcome was observed. In addition to the 4.2 inches reduced across the waist, hips, and thighs, patients displayed an additional circumferential loss of 3.5 inches along the non-treated regions. For the sake of time, we are unable to get into the detailed biochemistry of this secondary mechanism. But since the adipose tissue functions as an endocrine organ, we believe the volume reduction along the treatment sites modulates the hormones released, affecting systemic, non-Zerona treated adipocytes, promoting the release of their intracellular lipids. In addition to the 567 subject study, we also performed a 2,000 subject study evaluating the circumferential loss across the waist, hips, and thighs, subsequent to a standard Zerona procedure. In this 2,000 subject study, we demonstrated again a consistent result of 3.78 inches lost in 2 weeks' time.
One of the questions we often get is, why can I not simply diet and simply restrict the intake of high-calorie foods and achieve the similar response that Zerona is able to achieve in 2 weeks? The biochemical mechanism for lipolysis, which is the natural breakdown of triglycerides into non-esterified free fatty acids, is a complex biochemical cascade. In fact, along each enzyme, there are nearly 4 or 5 inhibitors that will immediately suppress this complex biochemical cascade. Therefore, no matter how much an individual can work out, if they do not adhere to an appropriate diet or lifestyle, the mechanism of action to break down triglycerides in the free fatty acids can be inhibited. Zerona skips this hormone-dependent biochemical cascade. We do not require the natural lipolysis of triglycerides in the free fatty acids. We achieve this outcome through mechanical means, through the absorption of photonic energy, liberation of the intracellular lipids, and the breakdown of triglycerides in the free fatty acids through the mechanism of action found within lymph nodes, lysosomal acid lipase. Therefore, Zerona is unique, able to achieve this significant and meaningful clinical outcome without going through the hormone-dependent, complicated biochemical cascade known as lipolysis.
In this image, we see the 3 transition steps of lipolysis, converting triglycerides into free fatty acids so that free fatty acids can be utilized as energy through a process known as beta oxidation. The blue spheres that we see, labeled 1, 2, and 3, represent regulatory steps. At any moment, if the body does not require the use of fatty material as energy, these steps will be inhibited. As discussed in the previous slide, step 1, lipolysis, is skipped by Zerona. Zerona will result in the increase in free fatty acids within the blood. However, we still must appreciate and respect steps 2 and steps 3, which is the breakdown of fatty acids into acetyl-CoA, which can be used as energy. If the individual does not have these important steps activated, the utilization of Zerona-liberated lipids will not be used as energy. Thus, we have developed a protocol in order to facilitate the highest degree of fatty acid metabolism, activating steps 2 and steps 3, which we will discuss a little bit later. An important aspect about adipocytes: when they are enlarged or hypertrophic, they can induce deleterious effects throughout the body. The image depicted here illustrates when fat cells are enlarged, we see an increase in the production or synthesis of low-density lipid proteins, LDLs, and the decrease in HDLs, high-density lipid proteins. This represents a prominent comorbidity for an individual who is obese or overweight. Here we see the direct relationship between fat cells and liver function. So we begin to understand how fat cells or adipocytes function as an endocrine organ cell. Adipocytes are endocrine organ cells responsible for the synthesis of bioactive substances known as adipokines. In a lean state, adipocytes secrete beneficial hormones including adiponectin, which actually promotes insulin sensitivity. However, as adipose tissue becomes enlarged or hypertrophic, we begin to see deleterious effects transpire. In fact, that beneficial hormone adiponectin begins to decrease in its concentration. Another hormone, leptin, which is responsible for the regulation of food intake and energy expenditure, begins to increase to a level where the hypothalamus no longer responds. Thus, we develop leptin resistance. In this hypertrophic state, adipose tissue is noxious and harmful to the body, altering glucose uptake, glycolysis, and gluconeogenesis. The purpose of Zerona is to take this enlarged state and revert it back to a healthy, lean state, seeing subtle changes in leptin secretion as well as adiponectin secretion. We continuously explore this medical utility, and we are currently participating in level 1 trials. But an important feature in this image illustrates that the endocrine organ function of adipocytes regulates the brain, muscle, and liver. Some of the secondary benefits observed subsequent to Zerona have been elucidated in numerous studies examining total cholesterol serum levels, specifically LDL levels and HDL levels. In this particular study, 80 subjects were evaluated, and after 2 weeks of treatment with Zerona, we were able to demonstrate a statistically significant reduction in total cholesterol. Furthermore, we were able to demonstrate a statistically significant reduction specifically in LDL levels, a 13% reduction in 2 weeks. It is important to note that for cholesterol-lowering drugs such as statins, the FDA requires at least a 15% reduction in 3 or 4 months. Here, we're demonstrating a 13% reduction in 2 weeks. Currently, we are undergoing a level 1 clinical study to evaluate the efficacy of Zerona for the reduction of cholesterol. This data is simply serving as a means to demonstrate the safety of this application. As we finish up on this slide, we see that HDL levels did not demonstrate a statistically significant reduction. In fact, numbers were preserved. Again, as fat cells become enlarged, we see an increase in LDL levels and a decrease in HDL levels. Restoring adipocytes back to the lean state would ultimately modulate that impaired pathway, maintaining or increasing HDL levels while decreasing unsafe LDL levels. Further studies demonstrate that following a Zerona treatment of 6 total sessions in 2 weeks, we are able to see again reductions in LDL levels, HDL levels, but more importantly, as we discussed earlier, enlarged fat cells augment the synthesis of beneficial hormones, which can give rise to comorbidities such as insulin resistance, otherwise known as type 2 diabetes. An important indicator for type 2 diabetes includes hemoglobin A1C levels and fasting glucose. In a separate study evaluating Zerona, we were not only able to demonstrate a reduction in hemoglobin A1C, but also a reduction in fasting glucose. Please recall that hemoglobin A1C recycles every 3 months. Thus, this study included a 9-month follow-up in order for us to properly evaluate changes in hemoglobin A1C. How is this all possible? Again, the fat cell is responsible for the synthesis of important bioactive substances, including leptin. In a separate study, we evaluated leptin levels and were able to produce a statistically significant reduction in leptin. Recall that an obese or overweight individual will present with higher levels of leptin. Therefore, by supplying a Zerona treatment and restoring a lean state, we have been able to document a reduction in leptin in 2 weeks' time. What does the overall procedure mean for you, the clinician? Because the secondary metabolism is so complex, and that the Zerona is simply representing a limited function, we strongly encourage that coupled with this process are adjunctive components. Again, there were no adjunctive components in our clinical study. We could make no suggestions regarding hydration, supplementation, or exercise. But now having FDA approval and understanding the complexity of the human metabolism, we can incorporate these components to facilitate greater fatty acid breakdown. When a patient first comes in for their initial consultation, they will not receive Zerona treatments that first week. What we encourage is that after that initial consultation, you send the patient home with supplements. They begin to consume those supplements in preparation for that first Zerona treatment 1 week from that day. In addition to the consumption of supplements, patients practice hydration techniques, simply consuming the American Medical Association's daily recommended consumption of 2 liters a day. With nearly 75% of Americans chronically dehydrated, it is important that patients try to maintain a proper hydration status during this process. After that 1-week preparatory session, patients will come in week 2 ready for their first Zerona treatment. They will receive treatment every other day, Monday, Wednesday, Friday, or Tuesday, Thursday, Saturday, for that first week and that second week of treatment. During that time, we ask patients continue to maintain hydration. Furthermore, we ask patients to participate in some type of lymphatic stimulation. You can present a menu of options for patients, but when understanding the mechanism of the lymphatic system, we know that it's an open vessel system. It does not rely on a pump-driven modality. In fact, it relies solely on muscle contracture, movement. And if we are liberating the lipids into the interstitial space, which is regulated by the lymphatic system, it is important to enhance this system through either compression garment, walking, even self-palpation while an individual sits at their desk, a massage. These are important strategies to help support the removal of the fatty material, and this will occur during weeks 2 and weeks 3. After the 6th and final Zerona treatment in week 2 of their treatment phase, the individual will not receive their last measurement. In fact, we encourage patients to return 1 week after their last Zerona treatment. The reason being, all we have done on that last treatment is liberated more fatty material. Therefore, we need a few days for that material to be removed and metabolized by the body. Thus, we ask the patient return 1 week after their last Zerona procedure in order to acquire an accurate measurement for the circumferential slimming. The protocol I just discussed was for a patient population between a 20 and 30 BMI, normal weight to overweight. The Zerona, though, as a diverse and unique application, has the capacity to treat individuals that extend beyond a 30 BMI, those individuals that are classified as obese or morbidly obese. Presently, 66% of the U.S. population is either overweight or obese, and it is estimated by 2030, 87% of our population will be overweight or obese. This will contribute to nearly $1.5 trillion of annual health care cost to simply control the comorbidities of this population. Therefore, Zerona can represent an alternative strategy to help promote a meaningful slimming for this population. A study performed by Dr. Capasso in Florida examined 88 subjects. In that study, he had a BMI range of 21 to 45, a normal weight individual to a morbidly obese individual. The treatment range included 6 to 18 treatments. Obviously, the individual presenting with a 21 BMI received 6 treatments, where the individual presenting with a 45 BMI received 18 treatments. The inch range in this clinical study was 3.75 inches to 26.8 inches across the waist, hips, and thighs. 3.75 inches is not meaningful for an obese individual, but 26.8 inches with simply an extension in the treatment time, that's meaningful. And that accurately reflects the diversity of this application. We are not restricted to the treatment for individuals that simply want a cosmetic adjustment. Within your practice, if you see patients that are struggling with their weight, that are looking for viable alternatives, please think of this as a viable strategy. A majority of individuals will fail to comply to dietary components simply because they lose that intrinsic motivation. If Zerona can be there to produce a circumferential reduction that can generate meaningful results, then it could potentially serve as that apparatus to promote or to sustain that intrinsic motivation. We mentioned in both protocols the consumption of a supplement. We can provide a supplement that contains key ingredients that can help promote fatty acid metabolism. As nearly 80% of individuals in the United States not receiving or consuming adequate supplements, minerals, or nutrients, it is imperative that we introduce a supplement to help support the biochemical breakdown of fatty material. It is not important that you purchase our supplement. It's important that you recognize that supplementation is an essential function. If you have a provider or currently provide supplements, that is a viable alternative. But what we have to understand is that L-carnitine is the sole transporter of fatty acid into the mitochondria to be used for energy. Without L-carnitine, we are unable to promote fatty acid oxidation. Moreover, niacin serves as a component of coenzymes 1 and 2 for NAD and NADP. These are imperative components for the breakdown of fatty material. If an individual is not consuming an adequate supply of niacin, we can have limited fatty breakdown. This supplement will not generate a slimming event. It will simply support the breakdown of the fatty material we're able to release by Zerona. The image we see here is the complex human metabolism as we know it. Thousands of biochemical reactions work synergistically to support homeostasis within a cell. Fatty acid metabolism and the release of fatty material via Zerona represents simply one line in this complex biochemical mechanism. When we see an image like this, and we respect the human metabolism, this is why we incorporate hydration, why we ask patients participate in some form of lymphatic stimulation, and why we strongly encourage the consumption of supplements. Again, we could not include any of these components in our level 1 clinical trial, but understanding this mechanism, understanding the human metabolism, we're able to enhance the results. In fact, the 70% success rate we were able to produce in the clinical study is now 93% in our retrospective studies. That is because in those studies we're able to recommend the consumption of 2 liters a day of water. We are able to recommend the consumption of a supplement. And at the end of the day, we see tremendous improvements in our overall results. Any non-invasive body contouring device should have an image of the human metabolism. Because if they do not understand this complex mechanism, then they are disregarding the important aspect regarding the utilization of fatty material for energy. As we come to the end of the science presentation, it is important that we reiterate our commitment to clinical investigation of the highest quality. Everything that we have discussed has been peer-reviewed published, starting from our histological evidence, which was peer-reviewed published in Plastic and Reconstructive Surgery Journal, to our level 1 in vivo study, which was published in Lasers in Surgery and Medicine. Earning peer-reviewed publication demonstrates that our peers accept our research and our work and find it acceptable for publication. so that other researchers have the ability to learn more about this process. We remain committed to the exploration of this technology from the cosmetic discipline to the medical discipline. We can see here a list of our publications. So please, at any time you would like to read the full article, do not hesitate to contact us, as we will provide you with the entire publication so that from the publication you can make your own decisions and you can see the proper parameters that we had to implement in order to properly elucidate the underlying efficacy and safety of this application.
This concludes our science presentation. Hopefully we were able to appropriately articulate the underlying mechanism of action that contributes to Zerona's ability to generate a significant and meaningful circumferential reduction in as little as 2 weeks. We discussed a significant amount of material in this presentation, and if you still have any questions regarding the procedure, please do not hesitate to contact your local sales representative or any Zerona associate. If you would like to also download any of the materials we discussed, please visit zeronasuccess.com. Again, thank you for your time.